Well, Joellen, thank you for a very, very stimulating start to our discussion. Could I invite Veronique, I can't see her, here she comes, onto the stage. Veronique Garcon is a senior scientist at the... Right, here you go. I'm afraid to lose you. Is a senior scientist at the Laboratory for Space Geophysics and Oceanography at Toulouse. I always get Toulouse and Toulon confused, to think for a moment, where she leads a similar group to Joellen's in the area of physical dynamics and biogeochemistry. She got her PhD at the University of Paris and then moved for a couple of years to Sally Chisholm's lab at MIT, then back to France, and then spent some time at Princeton University with Sarmiento. She's been engaged in work on the effect of large-scale dynamics on ocean biogeochemistry. And she's going to talk to us on what the very important issue, at least it would seem to me, of the changes in stratification on marine ecology and the changes that climate will induce on stratification. I think that's a more correct title for it. So, Veronique, thank you. And I will let you know when we're doing well for time.
Okay, first of all, it's an immense pleasure to be here today for this upcoming ASLO week in Nice. And I would like to warmly thank the organizers, the chair of this meeting, for inviting me for this plenary session. The title of my talk is dealing with biogeochemical cycles and marine ecosystem in a changing oceanic stratification. It's a rather wide topic, and as one can guess, it's almost impossible to cover it in 25 minutes' time. So I will rather provide a biased, non-exhaustive, personal view of the topic. And I built this presentation with the help of Dr. Paulmier, who is presently at the Max Planck Institute in Bremen, Germany. Brief outline of my talk. I'll say a few words of rationale, but I think Johanna already introduced it quite nicely. Then we'll see how marine ecosystems adapt and behave in a warmer world. A few words of the other CO2 problem, which is ocean acidification. And then we'll see the expansion of oxygen minimum underwater deserts. And I will say a few words on coastal hypoxia before short conclusion. So human society have been interacting with their environment spanning many millennia, and now there is a dawning awareness that the Earth itself is a single system in which biosphere, both terrestrial and oceanic, is an active component. And this 650,000 years record really provides a visual evidence of the fact that CO2 and climate have varied together in the past. And if we look at the CO2 record in red, methane in blue, and deuterium in black, which is a proxy for local temperature, we see that this pattern really exhibits the same dynamics and patterns over the entire record. And they also fall within the same bounding envelope over several interglacial cycles. So if we now put the present in perspective with the past 10,000 years or so, we see that the greenhouse gas concentration shown here for CO2, methane, and nitrous oxide have been increasing dramatically at a very unprecedented pace. For CO2, we're reaching now close to 384 ppm. So we know we have an observed trend towards a warmer world, and this is a consequence of greenhouse gas concentrations. These panels show the annual global mean temperature taken from the last IPCC report. Here the temperature, and we are looking at the trend over the past 150 years. The colored lines show the trends over the past 25, 50, 100, and 150 years. And over the last century, we have 0.74 plus or minus 0.2 degrees C warming. So, yes, the oceans are warming, and here shown the geographical distribution of the linear trend of change in ocean heat content in the top 700 meters of the water column. So we see the distribution is not uniform. We have region experiences warming, like in pink-red, and others cooling in blue. So in a warmer world, we have an observed increased atmospheric CO2. We have also observed change in physical property of the ocean, and these two have an impact on marine biogeochemical cycles. How? First, the increase in atmospheric CO2, of course, leads to additional CO2 being dissolved in the water column, leading to a change in the equilibrium in the oceans. Changes in temperature and salinity also have an impact on solubility and chemical equilibrium of the gases. And finally, the changes in circulation in the oceanic flow will have an impact on the supply of carbon and nutrients from the intermediate waters, on the ventilation of the oxygen-depleted waters, and also on the downward penetration of the anthropogenic carbon. So both physical change and biogeochemical change will in turn impact biological activity, which may feed back also on the biogeochemical cycles. So how does phytoplankton behave in a warmer world? We're looking here at a picture from Berenfeld et al. Paper in Nature 2006, where with a decade of satellite ocean color data, basically the Seaweeds archive, they reported that, in fact, the phytoplankton biomass and the growth were declining as the ocean water surface were warming up. We're looking here at a picture of net primary production derived from ocean color data, so high values with high nutrient surface concentrations, and the contour, the black line here, telling me the permanently stratified ocean where the annual average surface temperature is above 15 degrees C. So basically what they did was to compute the monthly anomalies of global depth-integrated chlorophyll concentration over the Seaweeds archive over time here, and these are the gray dots, the green lines, sorry. And we see that these anomalies are dominated by changes in the anomaly in the permanently stratified ocean region, where we have that annual average SST greater than 15 degrees C. We have the same kind of pattern for the NPP anomaly. Two slopes, two trends appear, a very ascending slope between 97 and 99, which correspond to the transition from El Niño to La Niña, and since 99 we have a drop in chlorophyll concentrations. So quite a clear evidence of climate plankton link. So how ocean stratification can affect marine productivity? In three ways. One, in setting the nutrient surface concentration by controlling mixing between the surface and deeper waters, by determining the length of the growing season, so the light available for photosynthesis, and in setting the general environmental condition. These three mechanisms can oppose each other, and the marine productivity can equally decrease or increase as a result. This is from a slide from Scott Donnay, where indeed in the tropical and mid-latitude where growth is nutrient limited, we have a reduced mixing because the water column is stabilized by thermal stratifications, and with warming we have a reduced mixing which limits the upward nutrient flux into the euphotic zone. Whereas for high latitudes where light is limiting and we have an intense vertical mixing which breaks phytoplankton cell down at depth, when we have warming and an increased input of fresh water due to increased precipitation and melting of sea ice, then we have a mixing which is diminished, and in that case would favor marine productivity. So what Berenfeld et al. Did, they kept that record of NPP, and for the stratified ocean, they defined a stratification index, which was the density difference between the surface temperature and the 200-meter depth. And they computed these monthly anomalies for the stratified ocean. And we see the red dots, of course, here. So we see that the two patterns really agree rather well, as an indication of a close link coupling between NPP and climate variability. Just to look at the global pictures, in the top, so red is positive and blue, purple is negative. We're looking at the SST changes, the NPP changes, and here the correspondence between the two. So if we look at that yellow area here, which it corresponds to a positive SST change associated to a negative NPP, and the blue here, a negative SST associated to a positive NPP. So they computed that for about 75% of the permanently stratified ocean, we have NPP which vary inversely with changes in SST. So not only phytoplankton is impacted, the higher trophic level can be also impacted because the recruitment success is highly dependent on the synchronization with pulse planktonic productions. So what Edwards and Richardson in their Nature paper in 2004 looked at, they tried to see whether there were climate warning signals emergent across the pelagic assemblage. And to look at that, they used the longest running marine biological survey in the global ocean, which is a continuous plankton recorder. And they looked back from the 1958 to 2002 period, and they looked at change in pelagic phenology across three trophic levels, primary, secondary, and tertiary production, using 5 functional groups. These gathered to 66 plankton taxa, and we are looking here at the North Sea ecosystem. So let's have a look at the top panels. What we look here, we're looking at 2 out of those 66 plankton taxa. Here is a diatom species, Cylindrotheca, and here is a dinoflagellate species, Terratium. What we look here, we look at the seasonal peak in production. So here we have the spring bloom. I forgot to say that they split the periods into 2 periods, before 80s and after the 80s. So here, if we look at the spring and autumn bloom, we see for the diatom there is not much change, quite stable indeed. Whereas for the dinoflagellate, we see that we have a forward in time peak for the latter period, after the 80s. This is the kind of same plot, maybe more visible, the months of seasonal peak over the 40 years record, spring bloom, autumn bloom, so we see fluctuation, oscillation, but pretty much stable. Whereas for dinoflagellate, we see a clear trend toward a forward in season for the peak of productions. So the intensity of the response will vary considerably among the different taxa of a trophic assemblage. And the different extent to which functional groups may move forward in time is leading to a mismatch between successive trophic levels, and also is leading to a change in synchrony of the timing between primary, secondary, and tertiary productions. And the bottom panel here is a kind of a sum up of all the 66 plankton taxa. So we're looking here at the change in seasonal peak from the whole 40-year period as compared to what it was in the first year of the record in 1958. This is a 0 line. When it's negative, means the organism peak earlier in season. When it's positive, later in season. So first thing to note, we have symbols everywhere, so there is not a universal trend. If we look at diatoms, which are the circles here, we see that we have some peaking earlier and some later. So pretty much consistent with this diatom species, but quite relatively stable. If we look on the dinoflagellate, the upward triangles, we see that most of the symbols are located here, meaning that they peak forward in time in the year. Same conclusion for copepods, and also same conclusion for meroplankton. So truly what it shows, at least in the North Sea ecosystem, is that the marine trophodynamics have been radically and profoundly altered. So, what's going on in a reduced pH ocean world? So we know rising atmospheric CO2 is tempered by oceanic uptake. But this oceanic uptake is not benign. It causes reduction in pH and alteration of the very fundamental chemical balances, the so-called ocean acidification. So we're looking here at a plot by Scott Donnay in a recent paper, We are at Station Aloha in the North Pacific subtropical gyre, and we see that the seawater CO2 and atmospheric CO2 record agree rather well, and indicating that the uptake of a tropogenic CO2 is a major cause for increasing DIC and a decrease in aragonite and calcite saturation state. If we look now at the blue dots here, which are pH measurements, which started in the early 90s, we see a reduction in pH of 0.02 units per decade. And this trend has been also observed at the Estoc and Bats Time Series Station in the Atlantic Ocean. So if we want to see what's going to happen in the future, this is a slide from Caldera and Wicked published in Nature 2003, where with a modeling approach, they used emission time series. It's a pretty old scenario. It's a mid-range IPCC 92 scenario, business as usual prior to 2100, and then assuming that the emission will continue until the fossil fuel reserve decline. So the top panel here shows the emissions record. The middle panel shows the resulting PCO2, which may reach 2,000 ppm around year 2300. And the bottom panel shows the vertical distribution in reduction in pH, so minus 0.1 for the next century, also up to minus 0.7 in the very far future. So all models kind of predict a dramatic decrease in pH in the ocean. So yes, the acidification is leading to a decrease in carbon and calcium in the ocean. And this is a picture from Ribezel et al. In this paper Nature 2000, where they looked at two representatives of Coccolitophorid, Geophyllocapsa and Emiliana, under a low CO2 world, 280 ppmv, and high CO2, 750 ppmv. And they reported reduced calcification rates, and we are looking here at scanning electron microscopy photographs, and we see in the middle for Emiliana that the coccoliths are malformed, and for Geophyllocapsa we see that the cocosphere are not complete. So this coccolith undercalcification and malformation is a common feature usually observed in natural environments and in laboratory conditions under high CO2 world. Scott Donay, in that same paper of review early this year, kind of gathered all the experiments that were carried out on the effect of increased CO2 on major groups of marine biota. And he looked at the calcification, photosynthesis, nitrogen fixation, and reproduction. These are the major groups listed, and 4 types of response. Linear negative, linear positive, at level, or nonlinear parabolic. So let's go to the table. If we look at calcification, we see that among all the species studied, many of the response exhibit a decrease in calcification rate. However, some strains of the same species may present a different behavior in different experiments. For photosynthesis, there are not so many species studied, but for those who were, they showed an increase in carbon fixation rate or a constant rate. For nitrogen fixation, the experiment was done on trichodesmium, and they experienced increase in nitrogen fixation rate. For reproduction, for the early developmental stage in benthic vertebrates, like in the echinoderms, we see that for all the study species, they showed reduced fertilization success and smaller developmental rates. So one word of caution is needed here, because all these experiments with high CO2 exposure are done for a few hours to several weeks, but nobody really knows what would be the long-term effect to a chronic exposure to high PCO2 and reduced pH. So another consequence of climate change is the expansion of the oxygen minimum layers. And we are looking here at a figure from a paper from Paulmier and Rispino published in PIO last year, where they kind of revisited the extent of these oxygen minimum layers in the modern oceans. And the extent may vary, of course, because of the reduced ventilation due to stratification, to the decreased solubility of oxygen, and also to fertilization, both anthropic and natural, which in turn impact on the remineralization rate. So we are looking at the top panel here, the oxygen distribution at depths where O2 is minimal. So that gives very clear where the OMZs are located in the eastern boundary upwelling regions, namely in the North Pacific, South Pacific. We have two major areas also in the northern Indian Ocean, Arabian Sea and Bay of Bengal, and to a lesser extent in the Atlantic Ocean, in the Mauritanian upwelling and in the Namibia. We see here that we are below 20 macromolar, so suboxic condition which can go to hypoxia on very coastal sites. So this is showing what was the extent, the estimation of the extent of this oxygen minimum layer when people were using a nitrate deficit criterion. And they superimposed the black contour here is the extension of these OMZ based on an oxygen criterion. Here they found 7% of the global ocean volume, whereas here it was 0.1. So quite a dramatic underestimation of the OMZ extension. At the same time, Strom et al. In Science 2008 looked at the tropical oceans. This is a map of oxygen, the purple region here, and we're going to look at the little square in all the oceans here. So the left panel, we're in the Atlantic Ocean. We're looking at the Mauritanian upwelling, Equatorial Atlantic, and Namibian upwelling. We're looking at 40 years of oxygen data here, the top 1,000 meter, and they kind of contour the 90 macromolar. So it's well above hypoxia, but it's a criterion. And we see that the thickness of that core, of the OMZ core, has been increasing, and also that the oxygen concentration is getting darker blue, so they are decreasing too. This is also true from this region here, but no trend is observed here. And instead, I'm not going to talk about the biological response to oxygen concentration, to low oxygen concentration in the open ocean, but I will rather say a word on coastal hypoxia. So this is a slide from a paper published by Duarte's group in PNAS last year, where they look at the hypoxia condition in coastal waters. And they show that the accumulative number of sites showing hypoxia, meaning oxygen concentration below which organism cannot live, has been increasing exponentially. So what they tried to look at is to examine the variability of the threshold concentration for marine benthic organisms in coastal waters. For doing so, they kind of gathered close to 900 experimental assessments where the oxygen concentration for lethal and sublethal were reported, and these amount to 206 species of marine benthic organisms. So we're looking at the top at the lethal concentration, sublethal threshold, and lethal time. First thing to note, there is, and these are classical box plots for all the organisms. So we see that there is not a single magic number, which was thought to be 2 milligrams of oxygen per liter, threshold below which all organisms would decide to die. So there is variety in response. And for instance, crustaceans, they are the most sensitive to low oxygen concentration because they present the highest lethal concentrations. Fishes have the highest sublethal threshold, and the most tolerant to very low oxygen concentration are gastropods and cnidarian, which show a very low sublethal threshold. So hypoxia impact occur at concentration of oxygen which are well above the 2 milligrams of oxygen per liter limit, and there is a wide variability in response. And this is important because these hypoxia have an impact on biodiversity. So in addition to oxygen depletion, we may have also occurrence of hydrogen sulfide to make things a bit more complicated. So this is a picture from a paper published in Nature this last December from the Max Planck group, where we're in the coastal Namibia and Pueling, and we're looking here at sulfide concentration in the bottom water, and here at sulfide at oxygen concentration. We see we have very low level of oxygen here. Here we are looking at a transect which is parallel to the coast, oxygen, sulfide, and nitrate. The top panel here shows the temporal occurrence of discoloration of surface water, which become turquoise, which you can detect with seaweed, for instance. So if we look at that middle panel here, at all stations where we have sulfidic bottom water, we see that there is no sulfide below the oxic zone. So what they did at 24 south was to look at the sequence of vertical concentration of hydrogen sulfide between the 15th and 18th of January, and they remarked a 15-meter drop in the sulfide front, despite the fact that sulfidic concentration were increasing in the very bottom water. So this apparent sulfide sink means that sulfide might be oxidized anaerobically with nitrate in the water column. And they looked at two sites at 24 south and 23 south. I just want you to look at here. So of course they measure anamox rates, which is anaerobic oxidation of ammonium. But here's the B plot, which is more interesting, and especially that elemental sulfur peak at 22 micromolar at about 87 meter depth. It's located right in the mixing zone between the nitrate in blue and sulfide here. And this is an indication of sulfide, of chemolithotrophic oxidation of sulfide with nitrate. And they reported cell density of proteobacteria, which are known to be involved in chemolithotrophic oxidation of sulfide with nitrate. So these proteobacteria in these fact are detoxifying the sulfidic waters. So nature can help sometimes. Time for conclusions. So we've seen the impact of rising CO2 biota, which can be varied with, there will be ecological winners and losers. In a warmer world, we've seen that, at least in our ecosystem, the marine trophodynamics may be really profoundly altered. The response of individual organisms, population, and communities to more realistic gradual change, long-term chronic exposure, is largely unknown. We've seen that, for instance, in some time, ocean acidification can lead to increased carbon fixation rate. So a more efficient biological carbon pump might lead to expansion also of these oxygen minimum layers. And these layers are truly key players to understand the imbalanced nitrogen cycle. And they're also key players for understanding the greenhouse gas control. I've said something about hypoxia because the coastal ecosystem are the most vulnerable, and it's the threat on marine biodiversity is really critical there. And so, of course, where to go? We need more laboratory mesocosm and long-term in situ monitoring, and together with the hierarchy of modeling approach to really see how changes in the metabolic process at the cell level will be manifested within ecosystem or community structure, and how in turn they will influence future climate feedback. And I couldn't prevent myself from adding these two lines, and this is a very personal view. And yes, we're facing climate change, but I think geoengineering solutions are not the solution. And truly, the ocean common do not need commercial apprentis sorciers. Thank you for your attention.